Welcome, everyone. My name is Anna Maria Nicodomska. I'm the Global Segment Market Manager for Public Health at Oxford Nanopore Technologies, and I'm delighted to welcome you all to this webinar today, where we'll be looking at how to use nanopore technology for viral respiratory pathogen surveillance. After the talks, we will have a live Q&A session, so please add your question to the Q&A box, which you'll find at the bottom of the screen. Where you can also upvote the questions that you would most like to be answered, and we'll try to get through as many as we can. The webinar will be recorded and will be made available on demand after the broadcast. So let's just jump right in and talk about what are the features that make our platform so powerful for the implementation of microbiology and infectious disease applications. The first is that our instruments are cost effective, they are accessible, they're easy to just put on a benchtop and use in almost any setting, whether it's in the field or in the lab. The second is how fast our turnaround time is, and this is enabled through both our rapid library prep as well as the generation of real-time data, which allows for real-time sequencing analysis as well. Because we're also sequence reads length agnostic, we can also generate reads of any length, which can be incredibly useful for the assembly of complicated genomes that have repetitive elements in them. So we can really break down our microbiology and infectious disease applications at Oxford Nanopore into 4 different sections. The first is microbial isolates, and we had a fantastic knowledge exchange on this a couple of weeks ago. You can just search for NOMIS. The second is our full-length 16S for microbial community profiling. And then we also have viral amplicon tiling for whole genome viral sequencing. And finally, metagenomic sequencing for agnostic pathogen detection. And so today we'll have 2 speakers who are going to focus on use cases for RSV viral amplicon tiling, as well as a second speaker who will be talking about metagenomic applications for unknown pathogen detection. So when would you want to take these targeted versus untargeted approaches for viral whole genome sequencing? Really, this is going to depend on your research objective, whether or not you know what you're looking for or not. And of course, there are several pros and cons that come with each of these approaches. With a targeted approach, you can't find novel viruses, and you can't characterize potential co-infection. However, you will end up with greater sensitivity, although possibly you may end up missing what else might be there. In terms of budget, this is also a consideration. Costs are generally lower for targeted approaches, and also computational resources tend to be lower. Whereas if you have a metagenomic sample, you might need to use host depletion approaches, and you also have a higher volume of data that you need to sift through to be able to find your pathogen of interest. So more complex data requires higher computational resources. So this slide just provides a quick overview of our viral whole genome sequencing solution for SARS-CoV-2. And this is a general approach that can be applied to other viruses as well. After we've confirmed our sample is positive for a virus, let's say through real-time PCR, we can reverse transcribe the RNA into cDNA, amplify our regions of interest through tiled amplicons based off of a reference genome, and then use that once we have those amplicons, pull them together, barcode them, attach our sequencing adapters, drop them into our flow cell sequence, and then analyze our data. And our end-to-end workflow for influenza genome is very similar, except that instead of using tiled amplicons, we will use primers at either end of each segment of the influenza genome so that you can generate a full-length segment for each of those 8 segments. We also provide bioinformatic workflows for both SARS-CoV-2 and flu that can be run through our epidemic software to analyze and give you human readable reports that contain things like viral typing, consensus sequencing, consensus genome of your virus, as well as potential typing. So what is our epidemic application? If you haven't used this already and you're not familiar with it, it's an open source graphical user interface that allows biologists who are not bioinformaticians, who don't have expertise in bioinformatics, to use these workflows to analyze their data. They can be run either in real time or after your sequencing run, either on your local device or in the cloud, and they will generate human readable reports as well as output files that can be used for further downstream applications. You can use either our locally developed workflows or community developed workflows, which Daniel will talk about in his talk next. And so with our final slide, we just summarize what we can offer in terms of sequencing solutions for viral whole genome sequencing, whether or not you're taking a targeted or untargeted approach, the sequencing kit that you'll need, third-party reagents, hands-on time, any other things that you might want to take into consideration before you make your decision to sequence. And so with that, I'm now going to hand over to Dr. Daniel Maloney, a research associate at the University of Edinburgh NHS Lothian, to discuss his work on RSV amplicon sequencing with Oxford Nanopore and what it reveals about both global and local lineage dynamics.
Great. Thank you very much, Anna, for that lovely introduction. And thank you very much for inviting me to tell you about our lovely amplicon-based RSV sequencing approach, as well as some of the data that we've generated so far with this method. Hopefully it'll whet your appetite for some future amplicon sequencing. So I've broadly broken this talk into three parts. First, I'll give you a quick intro into RSV and why people are so keen to sequence it at the moment. Then I'll talk a bit about our novel primer schemes and how they've been performing so far. And finally, fill you in on our sequencing efforts to catch the 2023-2024 RSV season in Edinburgh. So first off, RSV is a single-stranded RNA virus. There are two antigenically dissimilar subtypes, RSV-A and B, which share around 85% sequence similarity. There are no real major differences in disease phenotypes seen between the two subtypes, but one commonly dominates over the other during a given season. It's an incredibly common respiratory pathogen, with almost all children having been infected by the age of 3. And during the height of the season, RSV is one of the leading causes of admission into the pediatric ICU. It also seriously affects older populations, with around 750,000 hospitalizations annually. The RSV season typically starts just before the flu season each year, with it spreading in the winter months in temperate climates and during the rainy season in tropical climates. This was thrown completely out of whack, however, during the pandemic, with an almost non-existent RSV season in 2020. So before we move on, I'd just also like to quickly draw your attention to the diagram in the top right of the screen, and in particular to the cell surface protein encoded by the F gene. This is the fusion protein, which is vital for cell adherence and infection, and is a key target for current therapeutic interventions. And speaking of which, recently there has been a renewed interest in sequencing RSV at scale, as a number of countries have initiated vaccination programs. For example, in the UK, RSV immunization program has been underway since the start of September, focusing on two of the most at-risk groups, newborns and those over the age of 75. In the UK, both of these at-risk groups are being treated using the RSV pre-F vaccine developed by Pfizer. It is a bivalent vaccine made up of equal amounts of RSV-A and B stabilized pre-fusion F protein. And to protect newborns, RSV pre-F is being administered to expectant mothers a month or two before birth, and is hopefully not actually shown to scale here in this promotional material. So now that there's renewed focus on sequencing RSV in order to monitor how it changes as new pharmaceutical interventions come online, we felt there was a renewed need to be able to sequence it quickly, cheaply, and at scale. When we started this project in 2022, there were only a few thousand complete RSV genomes for each subtype. Part of the reason for this, we believed, was due to the existing methods at the time being somewhat labor intensive. The commonly used approach used 4 PCR fragments in 4 separate reactions, which has been very effective in getting whole genomes, but each of these 4 fragments needed to be independently quantified and pooled before sequencing could begin, which takes considerable time and effort in labs. To attempt to address this, we looked towards the success of the Arctic SARS-CoV-2 sequencing methodologies. In particular, the Arctic low-cost V3 library prep method for SARS-CoV-2 sequencing has worked really well and was used extensively during the pandemic, generating millions of SARS-CoV-2 genomes. As a result of this, there's a huge amount of lab infrastructure around the world ready to be used for sequencing, and we thought the best approach would be to design a 400 base pair amplicon scheme to match the SARS-CoV-2 current Arctic schemes. And this would allow labs who already use Arctic amplicon sequencing methods to slot these new primers directly into their sequencing workflows. To do this, we used the great primal scheme tool with a selection of RSV-A and RSV-B sequences to guide the design. This resulted in 2 primer schemes, one for RSV-A and one for RSV-B. For those not too familiar with amplicon sequencing, you can see a schematic representation of the 2 schemes at the bottom of the slide, with the genome going along the bottom and then the amplicons tiling along the top. Very briefly, if we zoom in, you'll see 6 gray amplicons that tile across this section of our genome of interest. And these are generated by a series of overlapping primer pairs shown as arrows in the diagram here. If all of these primer pairs were to be combined into one reaction, smaller fragments would be preferentially amplified, like, for example, this small section between the 2 red primers shown here. So in order to avoid this happening, you split the primers into 2 pools and amplify these separately. So here, all the orange primers will be in pool 1 and all the green primers will be in pool 2. And this will effectively generate all the odd amplicons, 1, 3, and 5 here, in one reaction, and then all of the even amplicons in a separate reaction. Then before you move on to your sequencing, you combine these 2 pools of all the amplicons into one single tube, and then you bring that forward for your sequencing library prep, and then hopefully we'll get some lovely whole genomes out the other end. So far, for our RSV-A and B primer schemes, our initial sequencing results look pretty promising. Here you can see results for both nanopore and Illumina workflows, where we essentially just swapped our RSV primers into our existing SARS-CoV-2 sequencing workflows. You can see RSV-A in red and RSV-B in blue in these graphs here. There are about 450 samples in each of these plots, and you can see that the vast majority of samples are achieving over 90% genome coverage, which is represented by the dashed line here in these graphs. The samples tested here were not selected based on their CT value, but the majority of them would be less than CT30, with those over that accounting for a decent proportion of the sequences that did not achieve the 90% coverage mark. An important point to note is that the nanopore data was produced with the older R9 flow cells and kit chemistry, and we hope to test the new kits in the near future, and hopefully we'll update the protocols once that has been achieved. As mentioned earlier, the F gene, which produces the F protein, the fusion protein on the surface of RSV, is an important target for vaccine development and efficacy, so sequencing it well is important for any would-be primer scheme. Thankfully, coverage of the F gene for all these samples was high. This graph here shows that the F gene completeness from both sequencing platforms was around 70 to 80%, or achieved around 70 to 80% complete F genomes across all samples. As a quick aside, since we've generated 2 separate RSV-A and B primer schemes, that means you are currently required to type your RSV sample before you attempt sequencing. To cut down on that time and expense, we've investigated multiplexing the RSV-A and B primer schemes together, and this seems to work quite well. However, there is a marginal decrease in genome coverage when the primers are multiplexed, opposed to just using the single correct primer scheme for a given sample. But in general, a strong sample will perform as well with the single or multiplexed version of these primer schemes. We currently only support the primer multiplexing on nanopore for reasons that I'm happy to chat about later, and have created a bioinformatics pipeline to handle these data. Our pipeline can be used on the command line or as part of the Epitome software platform. Either way, our pipeline can generate you some great consensus genomes using the Arctic Field Bioinformatics pipeline at its core. If you scan the QR code here, you will be taken to the repo there where you can see how it all works. Currently, we only generate you consensus genomes, but we're hoping to add extra QC features to the workflow in the future. So watch this space. So far, so good. But a primer scheme that only works in the lab it was developed is not much use. So we decided to see how well it works for others with their local RSV diversity. Since our initial virological post about these primers at the end of 2022, we received a fair bit of international interest in testing these primers out. And in total, 16 countries have gotten involved so far, which you can see highlighted in yellow across this map here. By the time we submitted our preprint, which you can scan down here to go and see, we had received sequences back from 8 other labs based in 5 different countries. The numbers here you can see are the RSV-A followed by RSV-B genomes that they have contributed to our preprint. The labs in question represent 3 in the USA, 1 in Ecuador, 1 in Ireland, 2 others in the UK, and 1 in Bangladesh. So we've got fairly good coverage across the globe here for sampling RSV diversity. For the most part, these primer schemes have worked really well in the hands of our collaborating labs, again, with the majority of samples having over the 90% coverage mark. And these external labs used a range of sequencing platforms, with 4 using nanopore sequencing technology and 4 others using Illumina sequencing methods. And most enacted a rough CT cutoff of about CT30 for their sample selection. Encouragingly, if we split the sample out by year of collection, you can see we get high genome coverage at least as far back as 2019. But we can still get genomes of over 90% complete as far back as 2015. And we hope that this is a positive indicator that our schemes will be able to cope with the increasing sequence diversity in the future as RSV continues to evolve. To visualize the diversity of our RSV samples that we've captured in our sequencing study, we built some maximum likelihood trees, which you can see here. We used all publicly available RSV sequences up to the start of 2023 season as the start of the 2023 season as background data. And you can see RSV-A on the left here and RSV-B on the right. Each of the colored dots represents one of the sequences generated either by us in Edinburgh or sent to us from our collaborating labs, and only sequences that achieved over 90% genome completeness were included in the analysis here. As you can see, the sequences in the study land all over the tree, and they suggest that our primer schemes are able to capture a wide range of the currently circulating diversity in both RSV-A and B at the moment. If you have a look at the legend, you'll see the colors assigned to each country, with blues being labs in the UK and reds representing labs in the USA. And it appears that there is quite a lot of geographical mixing of RSV genotypes, with sequences from each geographical area not really clustering particularly tightly with other sequences generated in that area. You can see here that in the RSV-B tree, for example. The widespread geographic diversity of RSV is a hypothesis that has had some weight added to it from a recent paper by Langendijk, where they demonstrated a large degree of phylogenetic mixing of RSV across countries, which appeared to be largely influenced by the strength of air travel links between different locations. Recently, there's been renewed efforts to create a unified lineage scheme to describe the diversity of RSV that is out there. And to address this, at the start of the year, a paper by Gauthier et al., which has now been published in Emerging Infectious Diseases, proposed a lineage scheme similar to that of the PANGO system used for SARS-CoV-2, but with a few key differences. Namely, the first letter indicates the RSV subtype, and the presence or absence of the letter D signifies whether or not a duplication in the G gene is present, something which has been pretty much ubiquitous since the early 2000s. Then the sequential numbers indicate the ancestry of the lineage. So rather conveniently for us, NextClade have implemented the new RSV lineage system into their website. So I assigned RSV lineages to the samples in our study, And this assigned three major lineages to our RSVA samples, AD1, AD3, and AD5, and three more to our RSVB samples, BD4.1, BD4.1.1, and BDE.1. Hopefully, you're still with me after all of that. And again, there doesn't appear to be any major country-specific lineages using this lineage scheme, with all countries in our study showing similar proportions of each lineage. For RSVA, there appears to be roughly equal proportions of all three clades present, AD1, 3, and 5. Whereas for RSVB, though, almost all samples within this study represent the BDE1 clade. This can be seen more clearly if you look at the proportion of lineages present across time in these samples, with there being a healthy mix of lineages present in the RSVA samples, whereas the RSVB samples show a more dominant lineage at any given time. If you take a step further back and assign lineages to all of the background sequences in the tree, including ours, you can still see that RSVA shows a lot more diversity in the range of lineages present, whereas RSVB tends to have these sweeps towards a single lineage at a given time. So next up, I'll outline some of our findings from the last RSV season. Up to this point, sequencing effort for RSV have typically sequenced a small number of cases for a given year, but we sought to sequence as large a proportion of RSV cases as we could for the 23-24 season in Edinburgh. As mentioned previously, seasonality patterns were thrown out of whack by the pandemic, but the 23-24 season, which you can see in red here, has pretty much returned to pre-pandemic structure. Compare this to the dark blue line for the 2021 season, which peaked earlier in the year compared to the previous season. In total, there are around 6,000 laboratory confirmed RSV cases in Scotland for the 23-24 season. If you're actually interested in having a look at more at this data, Public Health Scotland has got a great dashboard with all this information on it, where you can get up-to-date figures. And currently for this year, we're roughly on track with the 2019-2020 season, which you can see in lime green here. So at the Royal Infirmary, samples are only tested if a patient has respiratory symptoms, either on admission or if they develop later. This season, there are a total of 1,110 RSV positive swabs, and of these, around 830 had CT less than 30 and were sent on for sequencing. Of these, 559 were successfully sequenced, which is just over half of the positives received and approaching 10% of all RSV positives in Scotland. So those 1,110 swabs came from around 800 patients at the Royal Infirmary, and the solid green line here shows the admissions over the course of the season, and beneath it in light green, it shows the distribution of the collection dates for the 559 samples we sequenced, showing pretty good coverage for the entire season. On the right, we have the age and sex breakdown for the sequence samples, split into under 5s, over 65s, and in perhaps an unorthodox grouping, adults are classed as everyone else in between those two. By far and away, the under 5s are the largest group with almost 400 sequences, with about 75 sequences in each of the other groups. And you can see that there's roughly equal sex distribution between each of these groups as well. When we break down the sequences by age and look at what lineages are circulating in each group, you'll see that there's an almost remarkable similarity in the proportions of lineages circulating in each, with RSVA being the dominant season here in Scotland, of roughly 80% of cases being RSVA. ADE1 was the most prevalent lineage in each age group, and BDE1 was still dominant in the RSVB sequences. If you have a look at the density of sample collection dates over here on the right-hand side, there is a statistically significant shift in the distribution of cases between under 5s, with the median collection date being about 3 weeks earlier than the other two age groups. By both of these pieces of data tie in with the hypothesis that it is younger people with RSV who pass it on to their older family members and others throughout the course of the season. To have a little look at where our Edinburgh samples cluster with the global diversity, We built maximum likelihood trees of all publicly available RSV sequences from 2020 onwards, which you can see here. When we add in the Edinburgh samples in yellow, you can hopefully see that are much more defined clusters in the RSVA tree when compared to the RSVB tree. This data set is crying out for some proper phylogeography to be run on it, but unfortunately we haven't done that yet. However, some interesting preliminary insights can be gained if you highlight the other sequences from across the UK in the tree, which you can now see in blue. You'll notice some overlapping clusters with the Edinburgh samples. Not particularly surprising, of course, as we're part of the same landmass. But what I think is noteworthy is if you highlight the sequence from the Canary Islands in the tree in red, you'll see some strong clustering, particularly in the top of the RSVA tree here. And this is suggested of the strong travel links present between the two countries and ties back to the travel links being a significant source of spread, as was mentioned earlier. Another neat thing about the rapidity and flexibility of amplicon sequencing is that you can investigate potential hospital-acquired infection clusters. So we closely examined the phylogenetic trees produced in this study for sequences that derived from multiple patients but were tightly clustered together, typically ones that were only one SNP apart. And this gave us about 24 potential clusters in our cohort. However, if you filter these by having symptom onset at least 7 days after admission, that leaves no clear nosocomial clusters that we have detected at the Royal Infirmary. However, this was not the case everywhere, and we were contacted by our colleague, Dr. Tom Williams, and their colleagues at Guy's and St Thomas's Hospital in London, who had used our primer schemes to sequence a potential outbreak of RSVB. This cluster was identified by the Infection Prevention and Control Team at GST, as all the patients had been on the same ward and had been there for at least 2 weeks prior to the onset of their symptoms. In total, 5 patients were identified as being RSVB positive, and they all had samples sent for sequencing, along with a selection of other RSVB positive samples collected during the same time period from the hospital. The figure on the left shows a timeline of admissions and sample taking. When analyzed, however, you can see here on the subsection of the tree of the full RSVB tree on the right-hand side, that the sequences from the outbreak ward, shown in blue, all clustered together, whereas the sequences from elsewhere, only one is visible here, this red sequence, so don't cluster anywhere near these sequences from the same ward. Eagle-eyed viewers will notice that there are more than 5 blue dots in this cluster. And in fact, the sequences from the initially identified patients also clustered with those of 2 other patients who had been missed in the initial search. They were both residents of the same ward, but had been discharged before identification of the cluster and were only tested when attending as outpatients after they had developed symptoms. We're writing a report on this at the moment and believe that this is the first example of nanopore amplicon sequencing being used for an RSV outbreak. So in conclusion, we have some primer schemes that work well for RSV sequencing. The details can be found in our preprint, and there's another QR code for that on the next slide. But please get in touch there if you have any questions or thoughts on their use. We have also shown that in agreement with previous studies, there appears to be a diverse set of RSV lineages circulating globally at the moment, with travel links playing a key role in RSV lineage dynamics. There also appears to be little difference in the RSV circulating in different age groups, but there is a significant difference in the timing of peak cases. With that, I'd just like to say thanks to both of the teams that I'm part of, and in particular acknowledge the work of Gonzalo Fernandez, who's helped a huge amount with the design of the primer schemes and the testing of them here in Scotland. And also a huge thanks to all of our external collaborators who've tested these primers, and in particular Tom Williams and the team at GST for their help with the cluster. And finally, thank you very much for listening.
Thank you, Daniel. That was a great talk. And it's now time to hear from our second speaker in today's webinar, Dr. Nick Gauthier, who is a research program manager as well as a senior scientist at BugSeq. And he's going to be taking us through translating metagenomics from research to the clinic. Over to you, Nick.
Hi, I'd like to thank the ONT team for inviting me here to talk today. My name is Nick Gauthier. I'm a research program manager and senior scientist at BugSeq. And today I'm going to be talking about some work we've been performing to use BugSeq and ONT sequencing solutions to translate metagenomics from the research to the clinic and BugSeq's approach to doing so. I'll spend the first part of my talk today talking about some work we've been doing to develop, automate, and translate end-to-end respiratory metagenomics assays in clinical settings. And then I'll spend the last half of the talk talking about some work that Bugseek's been doing on the bioinformatics side to develop robust, accurate bioinformatic analyses for infectious disease and microbiology applications that are compatible with ONT sequencing workflows. And then I'll finish off by talking about some work we've been doing on the clinical interpretation side of piece that we see as one of the last miles towards translation for bioinformatics in the clinical metagenomics context. In terms of a background on respiratory pathogen diagnostics, the COVID-19 pandemic, among many things, I think really emphasized the need to develop robust and novel diagnostic strategies, where traditional approaches like immunological approaches, targeted molecular approaches such as RT-PCR or amplicon sequencing for surveillance are limited in that these approaches rely on a priori knowledge of a pathogen's genome. Whereas metagenomic sequencing offers the ability to have a pathogen-agnostic approach that can be deployed on day zero in the case of a emerging pandemic. We first started working in the respiratory metagenomics space along with collaborators at the University of British Columbia and Vancouver Coastal Health to develop an ONT metagenomic sequencing-based assay powered by Bugseek bioinformatic analysis to detect SARS-CoV-2 from nasopharyngeal swab samples. And through this pilot study, we found 100% specificity overall, 95.2% sensitivity for samples with a CT value less than 30, and then dropping off after 30. But we were really impressed by this approach that for a lot of the metagenomic positive samples, we were able to do things like reconstruct high quality genomes across the range of the SARS-CoV-2 reference genome, which enables use in public health applications for things like outbreak analyses, phylogenetic analyses, things like that. However, we did acknowledge some limitations through this assay that would hinder potential translation. And that included things like high cost, high end-to-end turnaround time that really would limit the use, and a lack of automated protocols that would really limit the use in clinical settings. So following that, a few years ago, we began working through a BARDA-funded project to develop an automated end-to-end nanopore-driven metagenomic sequencing workflow alongside those collaborators at the University of British Columbia, Vancouver Coastal Health, wherein we automated a workflow from extraction and sample prep through library preparation, including using the RapidSmart, an optimized version of the RapidSmart 9N protocol, and multiplexing of up to 12 samples per flow cell that enabled simultaneous processing of up to 60 samples at once on an ONT GridION sequencing device. Using the real-time sequencing capabilities of the Nanopore platform, we were able to reduce sequencing time to around 3 hours, and in real time perform host scrubbing on the device and direct upload to the Bugseek UI for bioinformatic analysis. And we were able to turn bioinformatic reports around consistently within 1 hour. That included pathogen detection, consensus calling of viral pathogens, things like AMR detection, among others, QC-related metrics within those reports. And in terms of how that looks for a seamless ONT and Bugseek compatible solution, users can drag and drop their raw sequence data directly from their ONT sequencer, in this case a GridION sequencing device, directly onto the onto the Bugseek user interface, web-based user interface. After dragging and dropping their FASTQ files, users can just name their analyses, specify the ONT platform and sequencing chemistry, and we provide optimized workflows for both the R9 and R10 chemistry that are compatible. specify the database in which we have both a curated reference database that I'll go into a little bit later in the talk, as well as support for the NTBI-NT database, which is more of a broad net. Users can specify their sample type that they're sequencing, as well as sequence material, being DNA or cDNA/RNA, and then just click upload and see the results appear with about an hour later. Through that BARDA-funded work, we performed a robust clinical trial, analytical and clinical trial. In terms of the analytical trial results, we found for RSV and SARS-CoV-2, we were able to hit limits of detection around 10 to the 3 copies per mil, and around 10 to the 4 copies per mil, so slightly lower for influenza, which is something that we're currently working to optimize. In terms of reproducibility, for run-to-run reproducibility, we saw a high positive percent, negative percent agreement between runs. And this also translated to high correlation of raw sequencing reads between runs as well. And then finally, as many upper respiratory specimens have high percentage of host nucleic acid levels within the samples, we found limited impact on high host nucleic acid levels on our ability to recover internal process control MS2 reads. We performed a clinical validation study using samples collected from Vancouver General Hospital on about 350 samples and found 100% specificity overall, 86.8% sensitivity for samples of CT less than 30. And this was also biased towards having slightly lower sensitivity, as mentioned, for influenza versus SARS-CoV-2 and RSV that had high sensitivity up until CT30 and beyond. And one of the useful aspects of using an internal process control, in this case, MS2 bacteriophage, with clinical metagenomics assays, is that we were able to normalize viral reads to internal process control reads and show that that had a strong linear correlation with absolute viral abundance determined from qPCR. Finally, one of the advantages to using this ONT-powered workflow was that it enabled us to tailor sequencing throughput to balance sensitivity with turnaround time. So when we ran the flow cells for the entire 48-hour duration and then performed a sensitivity analysis, we were able to show that that 3-hour sequencing turnaround time offered an optimal balance between sensitivity and turnaround time for clinical and diagnostic applications. But for cases such as public health surveillance, where you may want to prioritize high genome coverage, the sequencing run can continue for those less time-sensitive applications as well, really emphasizing the benefit of real-time sequencing using ONT. So just to summarize work from that project, I think we really showed that translation goes far beyond just raw accuracy, and processing thousands of diverse samples is needed across a range of pathogens. I think that was really highlighted where from our results differing between RSV and SARS-CoV-2 from influenza, wherein if you just validate it on one organism for these agnostic assays, it's difficult to generalize that across a range of different organisms or sample types for that matter. We showed that real-time ONT sequencing coupled with the drag and drop bioinformatics using BugSeq facilitates a streamlined workflow and rapid turnaround, and really highlighted the compatibility between BugSeq and ONT workflows. And I'll spend the next part of my talk talking about something that we realized, that developing, validating, and maintaining bioinformatic analyses in the context of clinical metagenomics is more research intensive than most would expect. So in terms of CLSI practices of quality assurance, we often get asked by users whether it's databases, whether it's analysis, whether it's versioning, what's most important in the context of validating bioinformatic workflows. And I think I'm going to spend the next few minutes emphasizing that all of these individual pieces are really just pieces of a broader bioinformatic puzzle, and each one is key to producing robust and accurate analyses in the context of clinical metagenomics. In terms of the metagenomic databases, there are obviously many major limitations to public databases. Taxonomic misannotation and RefSeq is prevalent, and you're trusting the data submitters in terms of the annotation and NCBINT, as well as the sequence data quality being submitted. In terms of over or underrepresentation, the 15 most represented sequences in RefSeq in terms of bacteria have as many base pairs as the remaining 28,000 combined. And there are over 34,000 E. coli genomes as of last July, whereas other bacterial species may have very few or no representation within RefSeq. Some of these results on our approach to validating and developing bioinformatic databases are highlighted in a recent publication by our CEO, Sam Trolton. So you're welcome to check out that publication to learn more about Bugsy's approach to developing robust databases. In terms of quantifying the problem, through that Barter work, we found that 61% of NCBI sequences annotated as bovine RSV were more similar to the human RSV reference than the bovine RSV reference. We also found that cross-kingdom contamination was pervasive within existing reference databases. And in this example, we found that influenza sequences were contaminated within the Comomonas falciparii reference genome, causing many influenza reads to get misannotated as Comomonas. Even within the human reference genome, it's been shown to be contaminated with bacterial sequences, which is critical in the context of respiratory metagenomics, where a large proportion of the nucleic acid within any given specimen, even with host depletion, is made up of a lot of human reads. Furthermore, there are many pathogens, rare or emerging, that are missing or underrepresented in RefSeq. And I've included two such examples of missing pathogens on the right there, but in addition, Plasmodium ovale, many acanthamoeba species, some of which are human pathogens, are not represented. And also several influenza strains are underrepresented as well, or missing within RefSeq. However, the contents of the reference databases aren't the only consideration, and in particular with regards to phages or plasmids that can have broad host ranges that could inform things such as AMR. That range is not captured in many reference databases. Another such example is for divergent species. So we had a Bugseq user recently sequencing carbapenemase producing outbreak by a US state. When they sequenced it and performed classification using a Kraken-based classifier, it identified their isolate as Providencia hoaxiensis, which itself was only described in 2019. However, when the analysis was performed using Bugseq, we identified it as a novel Providencia species using genus-specific average nucleotide identity thresholds in the analysis. And indeed, in February 2024, this paper on the right came out that identified 7 new Providencia species, including this outbreak taxon, highlighting that genus-specific nucleotide identity thresholds are key in terms of providing robust bioinformatic analysis, even beyond contents within a reference database. Reproducibility is obviously a key consideration, and we've been working with open source tool developers in order to identify issues with those tools. One such example is with metagenomic assembly that is inherently non-reproducible, and many of the common assemblers don't produce the same assembly with each invocation. And we've been working with developers of these open source tools in order to mitigate some of these limitations of metagenomic assembly. Furthermore, bioinformatic versioning aids in reproducibility and quality assurance. And it's really the combination of software, database, and pipeline versioning all together that provides clinical users with assurance in terms of the quality of their outputs for their given sequencing application. And locking down all three provides confidence in that quality. And then providing an output log to users to enable them to contextualize results or differences in results within a given time frame is key in the context of clinical metagenomics. So just to summarize, I think I've really highlighted that both the database and the analyses are essential priorities in the context of translating metagenomic assays into clinical settings. They're not mutually exclusive. And unless you're processing high volumes and capturing a wide diversity with feedback from multiple applications, it's very hard to detect when these changes occur. I'll spend the next couple minutes talking about some ongoing work that's currently being funded by BARDA to solve the clinical interpretation piece, which we see as the last mile in terms of translating clinical metagenomics into diagnostic settings. We see this as a really underemphasized area of study and wanted to answer the question, how do we get from metagenomic classification reports that contain a lot of tables, a lot of different summary metrics, and provide that in a format that allows clinicians to provide an accurate diagnosis without having to individually summarize or set thresholds for each individual application. And we think machine learning techniques can really aid in this dimensionality reduction of highly dimensional metagenomic sequence data. So how does that look in practice? In this example, you can see an example bug-seq report from a sterile fluid sample. And in this case, we have a structured listing of each taxa in the sample, their associated read counts, and different assembly and coverage statistics to provide users with contextualization as to the quality and the abundance of each individual taxa within a sample. So without having to look for a needle in a haystack, how can we take these metrics, such as read count, such as assembly length and quality, and summarize that in a way and use machine learning techniques to set thresholds to aid in clinical interpretation? So going from that previous report to something more like this, wherein you have an executive summary at the top identifying a potential pathogen in a sample, and the top right having QC status metrics on whether internal control or negative control thresholds are passing or failing to enable clinical users to have more confidence in the quality of the results. And then providing a similar structure microbial listing, but then going a step further to providing outputs akin to syndromic molecular diagnostic panels in terms of which pathogens were detected or not detected within a given sample type and specifying it to that given sample type. So we think that this kind of last mile to clinical interpretation is really key in terms of the translation of respiratory metagenomics. And I think throughout this presentation, highlighting compatible ONT and bug-seq workflows and how we can use long-read sequence data coupled with robust bioinformatics in order to push clinical metagenomics further towards translation. So with that, I'd like to acknowledge key collaborators at the University of British Columbia who performed the wet lab development for these end-to-end workflows that we're developing. Collaborators at the BC Genome Sciences Center enabled automation of those protocols, as well as clinical collaborators at Vancouver Coastal Health for providing samples to produce these studies, as well as collaborators at University of Oxford who collaborated with us on that pilot study. And also thanks to BARDA Drive for funding this work to enable translation towards clinical settings of respiratory metagenomics. And with that, I'll thank the ONT team for inviting me here to speak today about the compatibility of ONT and bug-seq workflows in the context of respiratory metagenomics. I welcome any questions, and if you have any further questions, feel free to reach out by email or check us out at bugseq.com to see what sort of applications and workflows we offer that are compatible with ONT workflows. Thanks a bunch.
Perfect, thank you. And so you can find out more information about today's broadcast using the QR codes, which you can find on your screen. This webinar, along with all of our previous broadcasts, are available to you to watch on demand. So please do visit the Oxford Nanopore website to check them out. You can also find more information about our upcoming webinars and knowledge exchanges on the Oxford Nanopore website. Finally, we would really appreciate if you give us feedback on this webinar. You can find a short survey on today's broadcast in the Q&A panel at the bottom of your screen. You can just click on the Polls tab, and this will remain open once the webinar ends. If you have any further questions about your projects or getting started with nanopore sequencing, please don't hesitate to contact us at support@nanoporetech.com. And thank you all once again, and please enjoy the rest of your day.